Now that we can identify the three main parts of a cell, we're going to take a closer look at each of the organelles and the structures in turn. But before I do that, I just wanted to point something out. So as cells are really small, they're the smallest living structure in the human body, they are very difficult to photograph. For that reason, in textbooks and other resources, they're often represented as an illustration. Now, depending on the resource or the textbook that you use, these illustrations are all going to look slightly different. The main structures and the layout will be the same, but there will be some definite differences depending on the resource that you use and the style of that particular illustrator. So in saying that, as we are moving through this content, please be mindful of these differences and try and use more than one representation of a cell when you're doing your revision. It's also important to keep this in mind, not just for cells, but for all of the topics that we cover in this unit. So the first structure that we're going to take a look at in a little bit more detail is the plasma membrane. And the plasma membrane is a flexible yet sturdy barrier that surrounds and contains the cytoplasm of a cell. The plasma membrane is made up of what we call a phospholipid bilayer, which is just two back-to-back layers of phospholipids. So bi meaning two, bilayer meaning two layers. So a single phospholipid, which is what we have here, is made up of a phosphate head, which is hydrophilic. So hydro meaning water, philic meaning loving. And two fatty acid tails, which are hydrophobic. So hydro again meaning water and phobic meaning fearing. These hydrophilic heads will orientate themselves on the outside of the plasma membrane so that they face the watery cytosol on the inside of the cell and then the watery extracellular fluid on the outside of the cell. The hydrophobic fatty acid tails will point inwards so that they're away from the water on either side of that membrane. Now, embedded within the plasma membrane or our phospholipid bilayer are several proteins which can be classified as either integral or peripheral proteins. So integral proteins extend into or through the phospholipid bilayer and are firmly embedded within it. Most integral proteins are transmembrane proteins, which means they span the entire length of that phospholipid bilayer, and they protrude into both the cytosol, or that fluid inside the cell, and the extracellular fluid. Peripheral proteins are then proteins which are attached to the phosphate heads or to other integral proteins at the inner or the outer surface of the plasma membrane. And these proteins aren't as firmly attached to the plasma membrane. Now, proteins have various functions within the plasma membrane. For example, some integral proteins form ion channels or pores, which are like little holes or tunnels in the membrane that allow specific ions to move through. Some proteins will act as a carrier, which selectively moves substances which can't pass through that phospholipid bilayer from one side of the membrane to another. Some proteins will act a little bit like a receptor, which will sit on the outside of that cell membrane and bind specific molecules that change the functioning of the cell in some way. And then some proteins are called glycoproteins, which are just proteins with carbohydrates attached to the end. These protrude out into our extracellular fluid. And the pattern of carbohydrate on these glycoproteins varies between cells. So they kind of act a bit like a molecular signature that allows the cells to recognize one another, as well as recognize a cell or an organism as foreign, so not being from within the body. So considering the functions of the plasma membrane, and the plasma membrane functions primarily to separate the internal and the external environment of the cell. It is selectively permeable, which means it permits the entry of substances into the cell, but not others. This helps establish and maintain that appropriate environment within the cell for normal cellular activities. The plasma membrane assists with the transportation of substances into and out of the cell, so through ion channels and carriers, as we just spoke about, also through endo and exocytosis, which we'll speak about in the next recording. And then thanks to those glycoproteins, which extend out into the extracellular fluid, the plasma membrane also plays a role in cell-to-cell recognition. Moving on to the cytosol, and remember the cytosol is the fluid part of the cytoplasm, often referred to as the intracellular fluid. It is a water-based solution in which organelles, proteins, and other cellular structures will actually float. Cytosol is made up of about 70% water and 20 to 30% proteins. This composition gives it a bit like a jelly consistency through which ions, organelles, and vesicles, which again we'll talk about in the next lecture recording, can freely move through. The functions of the cytosol are to provide structural support for organelles, those little mini organs that sit within the cytosol. The intracellular or the within cell transport of molecules. And then to act as a site of many chemical reactions which are required for a cell's existence. Taking a look at the various organelles now, and the first organelle that we'll focus on is the mitochondria. And because the mitochondria generate most of the ATP used within the body, and ATP is the energy of the body, mitochondria are often referred to as the powerhouse of the cell. So a cell may have as little as a few hundred or as many as several thousand mitochondria, depending on the activity level and therefore the energy requirements of that cell. So think a skin cell versus a muscle cell, for example. They're going to have very different energy requirements. And now a mitochondrion, which is a singular mitochondria, is a double membrane-bound organelle, which just means that it has an outer membrane and an inner membrane with a small fluid-filled space between them. The inner membrane contains a series of folds called cristae, which increases the surface area inside the mitochondrion, and this is where the reactions occur that produce most of the cell's ATP or energy. So taking a look at a mitochondria, these kind of funny looking shapes are our mitochondria. You can see the outer membrane and then the inner membrane. That inner membrane has all these folds called cristae, which is where it has the chemical reactions which produces our ATP. Now, interestingly, although the nucleus of each somatic cell, which is any of your cells except your reproductive cells, so basically any of your cells except for your sperm and your eggs, any of these somatic cells will contain genes from both your mother and your father, except for our mitochondrial genes, which are only inherited from your mother. So mitochondrial DNA can be used to trace your genetic history, but just on your mother's side. Next up, we have ribosomes. And the ribosomes are these pinky dots that you can see floating around here, as well as on this purple structure here. And our ribosomes are the site of protein synthesis or protein production. Some ribosomes are called rough ribosomes, and these are the ones that are attached to the extensively folded organelle, which we're going to talk about shortly. These ribosomes produce proteins that are destined for specific organelles, so they might go to other organelles within the cell. They might be inserted into the plasma membrane, or they may be transported out of the cell. The other organelles, which are just floating around in the cytosol, are called our free ribosomes. Free ribosomes produce proteins used within the cell. So you can call this used for domestic use. Now, the endoplasmic reticulum is this purple structure here, which we just mentioned. It's a network of flattened membranes that extend out from the nuclear envelope, which is the layer that surrounds our nucleus. The endoplasmic reticulum has two parts. It has the rough endoplasmic reticulum, which is these flattened sacs that have the ribosomes attached to them. And then it has a smooth endoplasmic reticulum, so for example here and here, which does not have those ribosomes attached to its surface. So proteins synthesized or produced by the ribosomes attached to the rough endoplasmic reticulum enter the spaces within the endoplasmic reticulum for processing and sorting. As we mentioned, these proteins are either transferred to other organelles, they're inserted into the plasma membrane, or they're exported via exocytosis. Because our smooth endoplasmic reticulum does not produce proteins, because it does not contain ribosomes, it produces fatty acids and steroids instead. Now, most of the proteins produced by the ribosomes attached to the rough endoplasmic reticulum are ultimately transported to different regions of the cell or to external to the cell. And the first step in that transportation process is through an organelle called our Golgi complex. You'll see here termed the Golgi apparatus. So the Golgi complex or Golgi apparatus are small, flattened, membranous sacs, which are often shaped in this kind of curve. It's located always in close proximity to the rough endoplasmic reticulum because it modifies, sorts, and packages the proteins that are produced in this rough endoplasmic reticulum. It packages these proteins into vesicles, which are like little circles within the cell, so they can be transported within that cytosol. We then have the nucleus, which we have mentioned before briefly. It's a spherical or a round oval shaped structure that's usually the most prominent feature of a cell. Most cells have a single nucleus, although some, like red blood cells, have none, and some will have multiple. A double membrane called the nuclear envelope separates the nucleus from the cytoplasm. This outer layer of the nuclear envelope is continuous with the rough endoplasmic reticulum. So it goes straight into that rough endoplasmic reticulum. Now, extending through that nuclear envelope, which I'll show you more closely on the next slide, are openings, which we call nuclear pores. Nuclear pores control the movement of substances between the nucleus and the cytoplasm. Inside the nucleus are one or more spherical bodies or circle-shaped structures called our nucleoli, or our nucleolus for a single. And these function in producing the ribosomes. Within the nucleus are also most of the cell's hereditary units, so our genes. These control the shape of our different cells and they direct the cells to act in a certain way. Our genes are arranged along chromosomes. So in our human somatic cells, which remember are all of our cells except our reproductive cells, we have 46 chromosomes, 23 from our mother, 23 from our father. Each chromosome is a long molecule of DNA that is coiled together with several proteins. This complex combination of DNA and proteins is called chromatin. So looking at some of those structures a little more closely, this whole structure here is our nucleus. It's surrounded by that double membrane, so you can see it's kind of got those two layers of our nuclear envelope. That outer layer is what then runs directly into our rough endoplasmic reticulum. These little holes here are what we call the nuclear pores. That's what allows the transport of substances from within the nucleus out into the cytosol or the cytoplasm. Within the nucleus are one or more nucleoli, or nucleolus as the singular. The nucleolus produces the ribosomes, which then exit the nucleus through those nuclear pores. Now, the main function of the nucleus is to contain our genetic material. Remember, our genes are arranged along chromosomes. Each chromosome is a long molecule of DNA and proteins. And this mixture of DNA and protein is called our chromatin.